The science fiction premise of mechanically editing the code of life is now an active clinical fact. Today, medical doctors can reach into a patient's cells and slice into the code responsible for genetic disease. For decades, genetic engineering was a guessing game. Researchers bombarded cells with radiation or harsh chemicals, hoping to induce useful mutations, but the process was unpredictable. Progress stalled because we lacked a microscopic scalpel. Researchers could identify errors in a sequence, but had no way to target one exact line of code without damaging the surrounding DNA. Rather than inventing a tool from scratch, researchers looked to a microscopic defense mechanism that has existed for billions of years. The discovery began with a 3-billion-year-old war. Every day, bacteria are besieged by invading viruses. When an invading virus penetrates the cell wall, it attempts to hijack the host's machinery to replicate itself. Bacteria defend themselves using CRISPR. This DNA sequence contains repeating blocks separated by unique segments, acting as a memory bank. Surviving an attack, the bacterium extracts a fragment of viral DNA and slots it into this array, cataloging the invader. This ancient immune memory proves that programmable data storage existed in biology long before humans ever built a computer. When a previously cataloged virus attempts a second invasion, the bacterium detects the familiar genetic signature and immediately triggers its defense system. The bacterium deploys an enzyme called Cas9. We can visualize this CRISPR-associated protein as a functional machine, a biological assassin acting as molecular scissors. First, the complex physically unzips the invading double helix, forcing the strands apart to expose the viral genetic code. Cas9 is a highly efficient executioner, but it is entirely blind. It cannot neutralize a threat without specific targeting instructions. Instructions are provided by guide RNA. The bacterium takes that genetic Mugshot and slots it into Cas9 to arm it. The armed Cas9 slides along the unzipped viral DNA, rapidly testing the invader's code against the guide RNA, searching for a perfect match. Once the exact match is confirmed, the protein snaps shut, neutralizing the threat by cleanly severing the virus's DNA. The combination of Cas9 and guide RNA creates a naturally occurring find and replace algorithm that operates with microscopic precision. In 2012, researchers Emmanuel Charpentier and Jennifer Doudna realized they could hijack this immune system. By swapping the viral guide RNA with custom genetic code, they could redirect the Cas9 scissors to any target in any living cell. We see the impact of this hack in the treatment of sickle cell anemia. Looking at these malformed red blood cells, the entire disease is caused by a simple broken letter among 3 billion letters of the human genome. Scientists program guide RNA to find that specific typo. Cas9 navigates to the location and cuts it out, allowing the cell's natural repair mechanisms to stitch healthy DNA back into the gap. By co-opting this bacterial system, we gained the ability to rewrite the instructions for human life with the same precision we use to edit a text document. Rewriting human biology offers the potential for medical cures, but it carries a rigid boundary that dictates its ethical weight. Currently, doctors focus on somatic editing. This involved modifying the cells of a living patient, like the bone marrow of a sickle cell patient, to cure an existing condition. In somatic editing, the genetic modifications stop with that individual. Because the reproductive cells are left untouched, these changes are never passed down to the patient's children. This makes somatic editing a localized medical intervention. It treats the patient without altering the overarching human species. The ethical debate intensifies when we consider crossing that biological boundary and moving from the individual patient to the human race itself. This is germline editing. It involves using CRISPR to modify human embryos or reproductive cells. Any genetic changes made here, whether removing a hereditary disease or engineering specific physical traits, are permanently written into the human gene pool. Those edits will be inherited by all future generations. We possess the mechanical ability to direct our own evolution, having lifted a microscopic mechanism from 3-billion-year-old bacteria. Now we must decide if we possess the wisdom to wield it.